Okay, listen carefully. For chapter 18, okay, this chapter feeds off of chapter 17, gene expression, except this chapter focuses on regulation of gene expression. In other words, the synonym for regulation is control. It'll primarily focus, okay, on the regulation of gene expression at the level of transcription. So that first step of gene expression. We'll start off by talking about gene regulation in prokaryotes, and then we'll transition into talking about gene regulation in eukaryotes. So again, this is why I put this slide down here, just to remind you, it doesn't matter if it's a prokaryotic cell or an organism that's a eukaryotic nature. Okay, gene expression is two steps, transcription and translation here. Where in this chapter, again, it'll focus primarily on the regulation of gene expression at the level of transcription. So you'll hear me talk some about RNA polymerase. I will mention some about translation, so that you do need to know this information, but again, the majority of it focuses on transcription. Now, I added a couple of video links for the information we're going to cover on prokaryotic gene expression. I just added it this morning. It's in both the Word doc and the PDF doc. Okay, so here it shows in general, okay, both all living organisms together, prokaryotic and eukaryotic, the regulation of gene expression can actually occur anywhere along the two-step process, even before or after, okay? In addition to that, if you notice, they're showing you here, instead of showing you what some people might describe as light switches, because a lot of times when you go over gene expression, the impression you're given is gene expression is like a light switch. It's either on and proteins eventually are produced, or gene expression is off. We're not getting the production of proteins in the end. But in actuality, it depends on the gene. Okay, if it's genes that code for, say, pancreatic hormones such as glucagon and insulin, you can think of those genes as being expressed in regards to a light switch. They're either on, gene expression is turned on, or gene expression is turned off. But there are other proteins, okay, as we'll talk about at the end, mainly in eukaryotic cells, where it's not an issue of on and off. In other words, gene expression of a certain gene is occurring, and it's just a matter of whether, okay, the rate of expression is at a high level, or whether there's some type of stimulus that leads to a decrease in gene expression. So often you'll hear gene expression is on, but it can be influenced. It can either increase or decrease. Therefore, they use these valves. You know, valves comparable to what you might have in your outdoor water faucet. Okay, so these valves, basically, if you turn it one way clockwise, that'll turn on, say, water flow through these pipes. Okay, but once you've turned it on, you can actually increase or decrease the rate of water flow. This is what we're talking about here. Some genes, it's a matter of they're on, but it's a matter of increasing or decreasing their expression. Now, when we get into prokaryotic gene regulation, we're not going to talk about it in terms of it being a water valve. We're actually going to talk about it in regards to it being more like a light switch. Either gene expression is on or off. As I said, this chapter focuses primarily on the regulation of gene expression at the level of transcription. So you have to know, what are the working parts we need for transcription? Well, we need our DNA template, which is going to be the gene, okay? And primarily what we need out of that parent double-stranded DNA is a reverse strand. That reverse strand will serve as a template to synthesize mRNA, which is showing you down here. However, we talked about how RNA polymerase, it's the enzyme that catalyzes this process called transcription. However, it was discovered that it cannot locate the gene on its own, which they refer to as a transcription unit, but this is a gene. It needs the help of a DNA sequence called the promoter to help it locate the gene, and not only locate it, but locate the start point of the gene. So it knows where to begin transcription or synthesizing the mRNA. They refer to it as an RNA transcript, but the same thing as mRNA. And we talked about how on the opposite side of the gene, okay, bracketing the gene, is another sequence called the terminator sequence to let RNA polymerase know, hey, you're finished with this process called transcription. You have made the mRNA correctly. You can stop now. So we're going to be at this level that we're talking about here, and we talked about how it consists of 3 steps: initiation, elongation, and termination. What we're going to be focusing on primarily when we go over prokaryotic gene expression is this part right here. How regulation of gene expression, not only what we're focusing on is at the level of transcription, but it has to do with whether or not RNA polymerase is being allowed or being inhibited from initiating transcription. So again, broke it up in 2 parts from here: prokaryotic gene expression, and then we'll talk about eukaryotic gene expression. So for prokaryotic gene expression, Okay, scientists discovered that some bacterial genes, okay, they don't exist in a one-to-one relationship. In other words, going back to this here, they don't exist in a one-to-one relationship. In other words, for some bacterial genes, you'll have the gene, okay, but it won't, and you'll have one promoter regulating that gene and a terminator sequence on the other side. But for some bacterial genes, you don't have that one-to-one relationship, just focusing on the promoter and the gene. Sometimes what you have, okay, is a single promoter, which is this here, this rectangle here, okay, regulating or being involved in the regulation of more than one gene, which they're showing you here that I'm circling here, okay. So we refer to this group of genes being regulated by one promoter, just focusing on DNA. We would refer to this group of genes being regulated by one promoter. We refer to this structure as an operon. Oh, that was bad. Okay, we refer to it as an operon. So what we're showing you here, when I'm circling the entire thing, this is our operon. And notice what they're showing you over here, I'll put it on this side, is what they've highlighted is that double-stranded helix. Again, I'm doing a terrible job. They look more like a B, trying to write a D. So what they're showing you here is the rectangles are actually highlighting stretches of DNA, that double-stranded helix. Okay, so here it says some bacterial genes were found to be grouped as operons, one promoter regulating the expression of multiple genes. What else they found out is where you have an operon consisting of one promoter and a cluster of genes or a group of genes, when transcription occurs, when RNA polymerase, which is shown here looking like a guitar pick, when RNA polymerase binds to the promoter and slides over to the first gene, it will actually transcribe each separate gene into one single molecule of mRNA. So again, the group of genes are transcribed as one mRNA. However, once this mRNA, prokaryotic mRNA, is transferred to the ribosome, prokaryotic ribosome, built into the mRNA, as they're showing you here with green and red, built into the mRNA are start and stop codons. So you can see here, there's a start codon here highlighted in green and a stop codon here, referring to this as one gene. Then we have a start codon here in green, referring to the beginning of a second gene, or transcription of a second gene, and we have a stop codon indicating this is the end, okay, of the genetic information here, and so on down the line. Therefore, when this one molecule of mRNA is transported to the ribosome recognizes that it actually contains multiple pieces of genetic information for separate polypeptides, okay, that either will form active proteins or come together forming quaternary active protein. Now, proteins from an operon, what they discovered is the reason why you find in prokaryotic organisms, or bacterial cells in this case, since most of the research or most of the discoveries were made in bacterial cells, what they found is that apparently the reason for finding these operons consisting of clusters of genes is because these genes code for proteins that are involved in the same cellular function. So when we talk about an operon, we're talking about DNA that consists of a group of genes. These group of genes code for proteins that are involved in the same cellular function, such as lactose metabolism. We will go over this. We will go over two specific types of operons. One of them is known as a lac operon. Okay, it contains three genes that code for three separate active proteins, which are enzymes that are involved in the metabolism of lactose. So we'll go over this more in depth. However, for an operon, let me get this on. Down here, for an operon, however, it doesn't just consist of a promoter, okay, and genes. Notice I put plural here, multiple genes. Okay, an operon also consists of another stretch of DNA sequence called an operator. Okay, listen carefully. Just like the promoter is a DNA regulatory sequence, just like the terminator is a DNA regulatory sequence, the operator is also a DNA regulatory sequence. So in total, what they're showing you are a total of 3 DNA regulatory sequences. They're not showing you over here, but there should be another rectangle here indicating there's a terminator sequence here. Okay. Now, the way these regulatory sequences work, well, terminator sequence, same function as we talked about in chapter 17. It just lets RNA polymerase know when to stop transcription. Okay. The promoter, still the same function. A promoter is required by RNA polymerase to help it locate the genes, but in the case of an operon, the promoter is required by RNA polymerase to locate the cluster of genes beginning with the first gene. The operator, however, the operator is bound by a different protein, as we'll talk about, called a repressor protein. Okay, we give this repressor protein special names depending on the type of operon we'll talk about, so I'll give you two examples as we go through this. But this repressor protein, okay, its job is to come and bind to the operator when it can under certain conditions. And when it binds to the operator, if you notice here, it'll actually interfere with or get in the way of RNA polymerase. So I can't write this out. I'm just going to try to write RP for repressor protein. Okay, try to write an R and a P for repressor protein here. Under certain conditions, and we'll talk about a couple of conditions, the repressor protein, okay, would bind to the operator here, okay, inhibiting RNA polymerase from being able to initiate and therefore perform transcription. So we're going to talk about this. So this protein here influences whether or not gene expression of an operon is occurring, and we'll start building on this as we go. Now, there are two classes or types of operons that have been discovered in primarily bacterial cells. Okay, there's types of operons that are referred to as inducible operons, and then there's another class or group or type of operons called repressible operons. Inducible operons. Well, the inducible comes from induce. If you induce something, you're making something happen. It's the simplest way to put it. Inducible operons, what occurs with these operons is normally gene expression of these operons is normally turned off. And there has to be some type of molecule present to help cause gene expression to turn on. We will go over a specific type of inducible operon known as a lac operon. So a lac operon is a specific type of operon that falls under or that is classified under an inducible operon. And we'll talk about how this italicized three-letter abbreviation, LAC, refers to the disaccharide carbohydrate lactose, which acts as the inducer molecule. Now we have second type of operons, which are our repressible operons. Okay, repressible operons, the opposite process in regards to how gene regulation occurs. Repressible operons, well, gene expression of these types of operons is normally turned on, and something has to occur in the environment surrounding this operon to cause gene expression to be turned off. We would go over a specific type of repressible operon known as a trip operon. Trip, okay, normally we use an uppercase T, but the trip here is in reference to the amino acid tryptophan, one of 20 amino acids in our body. Tryptophan. We'll talk about how some bacterial cells, such as E. coli, when there's not enough tryptophan in them, they actually have the ability to make this amino acid, synthesize this amino acid. But we will talk about how this amino acid trip, produced by the group of genes coded for by this operon, well, that same product trip, the amino acid, will actually be involved in turning off the expression of this operon. So we'll build on this as we go. So the bottom line is, okay, when we're talking about an inducible operon such as the lac operon, the inducible automatically tell yourself it's going to require a molecule that acts as an inducer to turn on gene expression. If we're talking about a repressible operon, that means it's going to require a molecule, okay, to help repress or turn off gene expression of that operon. So starting with the lac operon, what this figure here is doing is showing you the working parts of the operon. Now notice something, okay? Down here, it's telling you with the bracket that only what is underneath the bracket makes up part of the lac operon, okay? What's over here is not part of the lac operon. There, notice over here on this side, it's telling you what compound are we talking about when we use these different terms. So on the test, if you see LAC-I, promoter, or LAC-Z, LAC-Y, or LAC-A, or you hear me use the term operator, automatically tell yourself he's at the level of DNA. Okay. If you come down here and you hear me use terms such as repressor protein or beta-galactosidase, permease, or transacetylase, you have to tell yourself, well, he's at the level of the protein now. Okay, so you've got to, I've actually had issues with that, getting students to separate the two compounds so they don't confuse them. Now, if you look at the level of DNA, if we're going to talk about the lac operon, we start at the level of DNA. The operon itself is what is bracketed. It's just this stretch right here, and I'm going to expand it a little bit because they're not showing you the terminator sequence. Again, the lac operon. This operon specifically has its own promoter, which we talked about all operons do. It has an operator, which I mentioned all operons do. But specifically, it consists of a group or a cluster of 3 genes. The first gene is called the LAC-Z gene. Okay. The LAC-Z gene encodes or codes for a protein enzyme called beta-galactosidase. Okay. The LAC-Y gene, this encodes or contains genetic information for a protein enzyme called permease. And then the LAC-A gene, okay, it codes for or encodes for a protein enzyme called transacetylase. So they're showing you here whenever RNA polymerase can initiate and perform transcription, it will transcribe all 3 of these genes, the LAC-Z, LAC-Y, and LAC-A gene of the lac operon as one molecule of mRNA. But notice in the molecule of mRNA, here's a start codon to let the ribosome know this is the beginning of the LAC-Z gene. Here's a stop codon to let the ribosome know that's the end of the genetic information for beta-galactosidase. Then there's a second start codon in relation to the genetic information that was stored in LAC-Y and transcribed. And there's a second stop codon to let the ribosome know, okay, you've just finished translating the genetic information for the second protein enzyme, and then a third one from there. Now, why do we call this a lac operon? That's the purpose of this next figure. Okay. The reason why we call this operon the lac operon, well, it not only encodes for 3 enzymes, and these are the 3 enzymes, but these 3 enzymes are all involved in the same function. These 3 enzymes are involved in lactose metabolism, or the breakdown of lactose. Okay? So if you look here, lactose is a disaccharide. Well, when we talk about the lac operon, lactose is not only a disaccharide, but it's also going to serve as the inducer. It's going to be the molecule that actually initiates the expression of the lac operon genes, resulting in the production of these 3 enzymes, which are going to break lactose down, this disaccharide, into its simplest form, the monosaccharides glucose and galactose, so that the bacterial cell can use them for energy. Okay, thus we call it the lac operon again. So as I said before, when we're talking about an operon, such as the lac operon, it contains 3 genes that code for these 3 enzymes, and all 3 of these enzymes are involved in the metabolism of lactose. Now, presently, the way it works, beta-galactosidase, okay, is responsible, okay, for breaking down lactose into galactose and glucose. Permease is responsible for helping the bacterial cell absorb more lactose when it's present in the extracellular environment. Transacetylase, I put down on your notes, the function is unknown. They've actually discovered a lot over the last 15, 20 years, but I'm not going to give it to you because it's not nice and neat to explain what it does. Even I had to read it over a couple of times, okay? So just for now, transacetylase function is unknown right now, okay? These are the 2, but do know that transacetylase is one of the 3 enzymes involved, all right? Now, going back to this figure here, over here, they're showing you a gene called the LAC-I gene. The reason why they're showing you the LAC-I gene here is because the LAC-I gene codes for a repressor protein that's actually known as the, if I can do this, the LAC-I gene encodes or contains the genetic information for a repressor protein known as a LAC repressor protein. So this LAC repressor protein, if you look at it here, okay, remember, A protein's shape is directly related to its function. Okay, and this couldn't be truer than for a repressor protein. And they'll show you this. Okay, it's showing you here on the repressor protein that when it has this shape, this side up here, which looks like it has a couple of teeth, okay, this side of the repressor protein has a shape that fits neatly into this stretch of DNA. Well, it just so happens this stretch of DNA is the operator. So it's showing you here at the level of the protein, okay, whenever the lac repressor protein has this shape on one side of it, it has a shape that complements this stretch of DNA. Therefore, it can bind to this stretch of DNA, which they now refer to as a DNA regulatory sequence called the operator. But notice where the operator is positioned if we're just looking at the DNA. The operator is positioned between the promoter and the first of 3 genes of the lac operon. Therefore, if this protein has the correct shape and binds to the operator, it physically is inhibiting RNA polymerase from initiating and performing transcription. I'm pointing this out because this is what we're going to go into next. Now, often what I'll tell students is how this works. We're going to go over how this works. Okay, when we go over how this works, we break it up into 2 parts. When we go over how this works, we first start with when lactose is absent. Okay, down here before I get going any further, the lac repressor protein has a total of 2 binding sites. Okay, one of its binding sites, which is on this side, okay, binds to the operator. Okay, the other binding has, which they're showing you here with the curve, this binding site, this second binding site, is actually for lactose. Now, technically they call it allolactose, but I just want you to know it's lactose. Okay, is a binding site for lactose. Now, the way it works for this lac repressor protein is that both binding sites do not work at the same time. In other words, either this repressor, and I put the name over here because it just says active repressor, this repressor protein, the lac repressor protein, it is either binding to the operator or it is binding to lactose. It cannot bind to both. All right, so let me get going here. So when lactose is absent, the repressor, the lac repressor protein, is said to be active. Okay. And what that means is that it has a shape where the binding site for the operator complements the shape of the stretch of DNA that forms the operator. Therefore, when lactose is absent, the lac repressor protein is active and will bind to the operator. This results in the inhibition of RNA polymerase. RNA polymerase is physically being blocked from sliding over to the first gene, the LAC-Z gene. Okay. Therefore, in this case, the operon is off, or in other words, gene expression is turned off is the way we'll say it. Okay, so this is what I mean by the operon is off. Gene expression is off, or I like to say gene expression has been turned off. Okay, so please notice the stretch they're showing you here is the promoter, the operator, and LAC-Z. So when you look at this stretch, understand what they're showing you is just this part, the promoter, the operator, and a small piece of LAC-Z. And then they're showing you LAC-I on the left side. Okay, that is when lactose is absent. However, when a bacterial cell such as E. coli is exposed to lactose that's in the extracellular environment, some of that lactose can actually get into the cell. Is the best way, simplest way I can describe it. Not wholly totally accurate, but simpler. And what happens now, okay, is when some of that lactose enters the ICF of the bacterial cell, such as a eukaryotic cell, what occurs is some of that lactose acts as an inducer. So technically speaking, it is both a nutrient that'll be broken down and used as an energy source or to make other carbohydrates, but it's also what we call an inducer in relation to the lac operon. Okay, so you can see down here where it's showing you we have lactose present, and in this case, in regards to gene expression, it's acting as an inducer. Lactose is present. It's not showing it here, but it does not matter if the lac repressor, okay, is bound to the operator or if it's not bound to the operator. In either case, okay, the binding site for lactose is available. So when lactose is present, it will bind to the second site of the lac repressor protein, except this induces or causes a change in the shape of the lac repressor protein to where now the other binding site for the operator no longer has a shape that complements this operator. Therefore, the lac repressor is said to be inactive. And when the lac repressor is inactive, it cannot bind to the operator, and now RNA polymerase can initiate and perform the process called transcription. So in other words, the operon or gene expression of the operon is turned on. So it says operon on, but you can rewrite it as gene expression is turned on for the lac operon. Okay, but notice something I didn't point it out on the previous slide over here. Notice here, when there's no lactose present and the lac repressor binds to the operator, notice this second binding site for lactose, it still has a shape that complements the carbohydrate lactose. So this is what I mean. It doesn't matter if it's not or if it is binding to the operator. This second binding site is still available for lactose. However, when lactose is present and it is bound by the lac repressor, this other binding site is not available, and therefore the lac repressor cannot bind to the operator. Now gene expression of the lac operon is turned on by RNA polymerase. Okay, so this is what we mean by a lac operon being an inducible operon. It requires a molecule called lactose to induce the gene expression of this operon, turn it on. Are there any questions on that lac operon? Okay, now for the trip operon, this is the second operon that is a repressible operon. Okay, the repressible operon, it encodes for 5 polypeptides. In other words, it has 5 genes, which they show you here. Trip E, Trip D, Trip C, Trip B, Trip A gene. I will not test you on their names. What I want you to know for the trip operon is that it has 5 genes. Now for the lac operon, I do want you to know the names of the genes because that's a very popular operon that's discussed in your advanced classes. But for the trip operon, you don't hear as much about it, so just know that it consists of 5 genes that code for 5 polypeptides. And these 5 polypeptides form 3 active proteins, which are enzymes. And all 3 of these enzymes, which I show you here, enzyme 1, enzyme 2, enzyme 3, all 3 of these enzymes are involved in the same cellular process. That is the synthesis of the amino acid tryptophan. So tryptophan, which they also show you down here, is an amino acid. Okay. Now the way it works though, okay, is through what we call a negative feedback mechanism. Okay, that's how this trip operon works. So we call it a repressible operon because normally the expression of the 5 genes of this operon is on. Gene expression is turned on, and it has to be repressed or gene expression has to be turned off, which requires a certain molecule. So down here, okay, or over here, they're showing you the 5 specific names of the genes that make up the group of genes of the trip operon, and it's showing you that they code for 5 polypeptides that form 3 active proteins, which are enzymes. These 3 enzymes will take a precursor molecule, which I'm not going to test you on, just know it's a precursor, that eventually is converted through a series of chemical reactions into tryptophan, the amino acid. But what eventually happens, okay, is that the cell either will make enough tryptophan to where it doesn't need to make anymore, or let's say suddenly in the surrounding environment, tryptophan is available for the bacteria, so it doesn't need to keep making it. In either case, when tryptophan levels get too high, when the concentration levels get too high within the cell, that excess tryptophan will feed back in a negative manner. So the feedback mechanism is specifically negative feedback. And going this way on this side, what happens, okay, is that tryptophan, as we'll talk about, it acts as a co-repressor, activating a repressor protein known as a trip repressor, which will then turn off gene expression or repress gene expression. So we're going to talk about tryptophan is not only an amino acid, okay? It is also described as a co-repressor, which I have in your notes. So this is the actual tripoperon here. Everything's shown here, so please make sure you pay attention to the bracket up here. Now I'm not going to test you on this. Over here they actually give you the specific name of the gene that codes for the second type of repressor protein we're going to talk about, which is simply the TRP repressor protein. So you're responsible for knowing the name of the repressor protein, which is coded for by a different gene, but you're not responsible for knowing this gene. Over here they're showing you the tripoperon, okay? It's formed by a DNA regulatory sequence called the promoter, another DNA regulatory sequence called the operator, and a total of 5 genes. Now, as I said, I'm going to say it again. You don't have to know the specific names of these genes. Just know it's 5 genes, a cluster of 5 genes. Okay. Now, you can see here for the tripoperon, they're showing you each of these genes codes for a polypeptide. And they're all, when transcription occurs, when RNA polymerase is able to initiate and perform transcription, it'll transcribe all 5 genes into one molecule of mRNA. But you can see, just like with the lac-operon, the tripoperon, the mRNA produced consists of multiple start and stop codons, so that when this mRNA is transported to the prokaryotic ribosome, the ribosome realizes, hey, look, I'm going to make a total of, or synthesize a total of 5 polypeptides. Okay, but what they're not showing you here, but they are saying here, is that for these 5 polypeptides, the polypeptides coded for by these 2 genes will come together forming a quaternary active protein enzyme. The polypeptides coded for by these 2 genes will come together forming a separate or a second quaternary protein enzyme. And then one of the polypeptides directly folds into the enzyme. That I do want you to know. Just know that of these 5 polypeptides, okay, 4 of them are subunits that form 2 separate active enzymes, and one of the polypeptides forms just an individual enzyme. Now, getting into the main part, and it's going to follow along like with the lac-operon, except we're coming from the opposite direction. Okay, so if you look down here, when we go over this operon, unlike the lac-operon where we begin with lactose is absent, with the tripoperon, it's a repressible operon. So gene expression is on. That's where we start. Well, why is gene expression on? One of the reasons, or the main reason, is tryptophan is absent. Now, the amino acid tryptophan goes by a 3-letter abbreviation. Uppercase T, RP, which I gave you on the previous slide. Okay? We use a lowercase t, an italicized TRP, for the name of the DNA sequences that form the tripoperon. Okay? The uppercase T is referring to the amino acid tryptophan used to make proteins. Now, the way it works for the tripoperon, which is a repressible operon, is normally gene expression is on. Why is gene expression on? Well, first of all, tryptophan is absent. And when tryptophan is absent, that results in the repressor protein being inactive. So here's our inactive repressor protein, specifically called the TRP repressor protein, which I put here. Well, if you come down here, the TRP repressor protein also has 2 binding sites. One of the binding sites is for the operator, but the second binding site is for the co-repressor. Well, the co-repressor happens to be the same molecule that these 5 genes that code for 3 enzymes are responsible for synthesizing tryptophan. So the product produced by the 3 enzymes coded for by the tripoperon, that same product is also what functions, tryptophan, the amino acid. That same product is also the product that functions as a co-repressor. So in a manner of speaking, think of it this way. The amino acid tryptophan, symbolized by an uppercase TRP, it has a dual role. It's being made by the bacterial cell because it's needed to synthesize polypeptides and make active proteins. But at the same time, that same amino acid acts as a repressor, shutting off gene expression. Okay? But when there's not enough tryptophan present, when tryptophan is absent, the repressor is inactive. They're showing you the shape of it. Why is it inactive? Well, the first binding site binds to the operator. The second binding site binds to the co-repressor. But when the co-repressor tryptophan is not present, that affects this other binding site, the binding site for the operator. When the co-repressor tryptophan is not present, the trip repressor protein does not have a shape on the other side. Okay, does not have a shape on the other binding site that complements this DNA regulatory sequence called the operator. Therefore, the trip repressor protein cannot bind to the operator. This is normally, okay, the state that this repressor protein is in. It's usually in an inactive state. Therefore, normally RNA polymerase freely can initiate and perform transcription of these 5 genes, which eventually leads to the production of 3 active protein enzymes. Okay? However, to control the levels of tryptophan in a cell, because the cell needs tryptophan to help it make proteins, but it's one of those situations. Too little is not good enough. Too much is also not good enough, and that's the case in tryptophan. The cell wants to be able to regulate how much tryptophan is produced. I only need a certain amount. I don't want too little or too much. So now what happens, okay, is eventually either provided in the surrounding environment and taken up by the bacteria, or in this case, eventually enough tryptophan has been synthesized by the 3 enzymes. So let me go back here first. So eventually, let's approach it from this direction. Eventually, the 3 enzymes synthesize more than enough tryptophan. The concentration gets too high. So what happens now is that some of this tryptophan is involved in what we call a negative feedback mechanism to turn off gene expression of the trip operon. Okay, and that's what they're showing you over here. Okay, so this figure right here that they're showing you here, first of all, it consists of this stretch right up to just here at the beginning of the TRPE gene, except they're not telling you TRPE gene. It's just up to here. They're just focusing on the main parts, in this part here. This they're showing you again is not part of the operon. It's the gene that codes for the repressor. This is the part we're interested in over here. We have our promoter, we have our operator. Okay. Now, once the cell has synthesized enough of the amino acid tryptophan, or when enough tryptophan is present, what happens is the excess tryptophan that the cell is not using to make polypeptides ends up functioning as a co-repressor. Okay. And when it functions as a co-repressor, it ends up activating the trip repressor protein. So here they're showing you tryptophan. They're showing it as 2 rings because if you were to go back to chapter 5 and look at this amino acid, you would see its overall structure, and its R group especially. It has 2 what they call aromatic rings, or ring-like structures. Okay, so what they're showing you here is the excess tryptophan that has been made, or when tryptophan is present, that tryptophan now is not being used to make polypeptides. Instead, the tryptophan is functioning as a co-repressor. And what happens is this tryptophan here will bind to the trip repressor protein, taking it from an inactive state to an active state. So what I have in parentheses, let me erase that. So what I have in parentheses is actually pointing towards this red guy here, which is the trip repressor protein. So in order for this repressor protein called the trip repressor protein, in order for it to be able to inhibit RNA polymerase, it first needs to bind to tryptophan, the amino acid. So when there's extra tryptophan, it'll bind to the trip repressor protein, changing the trip repressor protein shape to where now the other binding site, this other binding site for the operator, now it has a shape that complements this DNA regulatory sequence called the operator, and thus it binds to the operator. So notice something. For the trip repressor protein, both binding sites will work at the same time. whenever gene expression of the TRP operon is being turned off. So again, for the TRP repressor protein, both binding sites will work at the same time whenever gene expression of the TRP operon is being turned off or repressed. So again, the excess tryptophan acts as a co-repressor. Why? Because it binds to and changes the shape of the TRP repressor protein to where now its other binding site complements the shape of the operator. Now this TRP repressor protein on one side is binding tryptophan, on the other side it's binding the operator, and on the side it's binding the operator, it is now inhibiting RNA polymerase. So now the TRP operon goes from gene expression being turned on to gene expression being turned off or repressed. Okay. So you'll see this for cells, and we talked about it back in, I believe it was chapter, I forgot which chapter it was, where we talked about the way cells can control enzymes. I'm sorry, chapter 8, self-regulation. Okay. All right, that's it for prokaryotic gene expression. What I want to get into now is eukaryotic gene expression. Okay. For eukaryotic gene expression, okay, definitely eukaryotic gene expression can be controlled at any point in the process. Okay, and more so than what you see with prokaryotic cells. The belief for prokaryotic cells is they've evolved to a point they're very efficient. Their genetic information is stored in a manner that they can, that it's, how do you say, a gene expression when it occurs, it's a very efficient process. That's why you have those operons. Hey, if I need beta-galactosidase, the bacterial cell knows that I need permease and transacetylase, so let me just go ahead and express all three at the same time and do that by having them under the control of one promoter. That doesn't happen in eukaryotes. Okay. The genetic information stored in eukaryotes, okay, it's a one-to-one relationship. I've never seen different. For every gene that's been identified in the human genome, it is controlled or regulated by one promoter. And on the opposite end is a terminator sequence. Okay. However, what they found, though, is that where the regulation becomes more, how do you say, where there's an additional regulation occurs, okay, it can happen during transcription or during translation. So regulation or control can happen at any point in the process, and therefore it's been found to be much more complex. Okay, gene expression itself is much more complex in eukaryotic cells, therefore the regulation of it is much more complex than what you see in prokaryotic cells. But we are more complex organisms. We're multicellular organisms. Okay, so far, as far as we know, we've been able to do a whole lot more than prokaryotic organisms such as bacterial cells. So if you look here, regulation of gene expression. It's a process that has been found to be essential for cell differentiation. If you recall, when a sperm fertilizes an egg, okay, that fertilized egg will transform into a diploid zygote cell. It has 23 pairs of chromosomes that all contain genetic information from mom and dad. And then that one zygote will go through rapid rounds of cell division involving mitosis, forming an embryo, which I often tell students to just think of, okay, as a ball of cells, identical cells. Okay, and that's loosely not 100% exact, but think of it as a ball of identical cells that all have come from or derived from that zygote. So whatever genetic information that zygote has, every cell that forms that multicellular embryo is also going to have. But for reasons not understood, but that is known is tied into the regulation of gene expression, the cells that form the embryo begin to differentiate. Okay, they start to take on special shapes and special functions. And researchers look at it and they're still trying to figure out, okay, well, how does this cell know to eventually become a cell that forms your bone, whereas this cell all of a sudden knows to become a cell that forms one of your skeletal muscles. Okay, now it's showing you here. When we talk about cell differentiation, now we're talking about differences between cell types results in differential gene expression, which falls under regulation of gene expression. The expression of different genes by cells with the same genome. So differential gene expression is the expression of different genes by cells with the same genome, okay, but it's tied into regulation. So it's showing you here, okay, we have an embryonic precursor cell. For some reason, somehow, some way, it's the only type of embryonic cell out of a group of them that is able to activate, okay, a muscle gene known as MyoD. Myo means muscle. When it activates that gene, we get gene expression and we end up with a protein called MyoD. This protein acts as a transcription factor. If you recall transcription factors so far that we've talked about, we talked about a group of transcription factors that help RNA polymerase locate the promoter in eukaryotic cells. Well, we're going to talk about additional ones as we go through this. Okay, this MyoD protein that is a transcription factor will help RNA polymerase locate other muscle protein genes. Okay, and therefore, eventually, this one out of many embryonic cells ends up expressing a group of muscle proteins such as myosin, and therefore, it takes on a special shape, a fiber-like shape, okay, but it is a muscle cell, and it has the ability to contract due to muscle proteins such as myosin, in which they haven't shown you here, is also active. Now, why one type of embryonic cell will differentiate into a muscle cell, such as a skeletal muscle cell or muscle fiber, and others will differentiate into other cells, they're not sure how, but regulation is involved. And in eukaryotic cells, that regulation of gene expression has been found also to be tied in to the different types of transcription factors produced by eukaryotic cells. Okay. Now, so in other words, we are going over the basics. Even in advanced classes, rarely do they go over different types of transcription factors. And even in grad school, you would have to go specifically into working with DNA and gene expression to come across the different types. Okay, but I will give you two groups. Now, in eukaryotic cells, what they've discovered, okay, one, unlike with prokaryotic cells where DNA is circular, in eukaryotic cells, DNA is described as linear. It has open ends. Also, what they discovered for eukaryotic DNA is that it's packaged. Okay, in other words, it's been found to be combined with a group of proteins known as histones. Okay, so it says here, the packaging and unpackaging of DNA also contributes to gene regulation. And what they're referring to by the packaging and unpackaging of DNA is allowing DNA to associate and form a complex with these histones, or through certain chemical reactions or cellular processes, pulling the DNA away from this group of proteins so that a cell can express a particular gene that happens to be covered up by these histones. Okay. Now, the way it works, okay, if you look here, they're showing you a sister chromatid, and what they do is they pull on the sister chromatid to show you the primary compound that forms that sister chromatid is DNA. We talked about this before in chapters 12 and 13. When we talked about cell division, we talked about sister chromatids. An original chromosome attached its centromere to a copy of itself, an identical copy. The main compound that forms it, however, I told you was DNA. But I lied to you. Technically speaking, okay, whether it's sister chromatids or chromosomes, the DNA is found, okay, wrapped around groups of histones, okay, that we refer to as, histone octamers. Okay. And what they found is that this DNA, it doesn't wrap one time around a group of histone octamers. You find it wrapping repeatedly around groups of histone octamers. Okay. So wherever you see DNA wrapped around a group of 8 histones or a histone octamer, we call that structure a nucleosome. So notice they're showing you here. Okay, they bracketed it here. This is a nucleosome, and then what they do is they pull the DNA away to release these little purple beads representing these proteins called histone. But if you go the other way, notice, okay, DNA is made up of multiple histones. And technically speaking, you will see these nucleosomes, whether the DNA tightly packs together into a chromosome or sister chromatids, or loosely packs or loosely coils forming chromatid. You will find the DNA wrapped around these histones. Now, in between these structures called nucleosomes, where we have DNA wrapped around a histone octamer, there is some unwrapped DNA. Okay. At one time, they used to call this unwrapped DNA linker DNA. All right. So I don't know if you can make it out here, but they're kind of showing you a small little piece here, right in here. You can see here it's not wrapped. It's not part of the nucleosome, which is DNA wrapped around a histone octamer. They used to call this linker DNA. And the initial belief, okay, was that any genetic information contained in the DNA of a eukaryotic cell would be located in this region called linker DNA, or where DNA was unwrapped. But with advancements in lab technology, scientists discovered, boy, we are way, way, way wrong. Okay, you can have genetic information stored in DNA that is also forming part of this nucleosome. Therefore, this is what I mean by the packaging and unpackaging can also contribute to DNA gene regulation of genes in DNA. Sometimes the DNA does have to be pulled away or unwrapped, okay, to get it away from these histones to express a gene. So that's one way gene regulation can occur, is just by the way DNA is packaged and unpackaged. Second way gene regulation, okay, is influenced is through transcription factors, okay? First things first, if you hear the term transcription factors, keep telling yourself we're talking about proteins. Okay. And what is the function of these proteins? Well, it comes in the first part of this name. Proteins called transcription factors, they are influencing a process called transcription. Okay, but the thing is, we just said RNA polymerase, which is shown here as a guitar pick. We said RNA polymerase is actually responsible for catalyzing transcription. Yeah, but we also talked about in eukaryotic cells, first of all, RNA polymerase can't even locate the promoter without the help of transcription factors to help it locate it. But in addition to those transcription factors that help RNA polymerase locate the promoter, there are the two other groups, okay, of transcription factors that also have an influence, okay, on RNA polymerase. Some transcription factors, and I'm not going to try to write this out, some transcription factors are called activators, okay. The group of transcription factors that are called activators, they enhance, okay, or increase the rate of RNA polymerase activity. So transcription factors that are called activators, they enhance or increase the rate of RNA polymerase activity. But there's another group of transcription factors, and we actually talked about them because you do find them in prokaryotes, but you find them in eukaryotes. There's another group or type of or class of transcription factors. They are called repressors. Okay? And just like in prokaryotes, these eukaryotic transcription factors called repressors, they decrease or completely inhibit RNA polymerase activity. So again, these eukaryotic transcription factors called repressors, okay, they decrease or completely inhibit RNA polymerase activity. So in other words, transcription factors called activators eventually cause an increase in the concentration of protein being produced by a cell, but they do it through RNA polymerase. And then the other group of transcription factors in eukaryotes called repressors, they indirectly cause a decrease in the amount of protein being produced, or they stop any more protein from being produced, but they also do it indirectly through RNA polymerase. Now, the way they're able to do it is that transcription factors can do it in multiple ways. One way they do it is through DNA. Okay. Up here it's showing you the term enhancer. Okay. An enhancer is a DNA regulatory sequence, okay, that you will find near a promoter. This sequence, called a DNA regulatory sequence, it will not be recognized and bound by RNA polymerase. Okay. DNA regulatory sequences called enhancers are recognized and bound by transcription factors, okay, called activators. So that's what I'm showing you here. Each of these little yellow guys that looks like they have, I don't know, ears to me or whatever. Okay, each of these little guys represents a transcription factor called an activator. All right, and notice they're all binding to these yellow little strips here that are highlighting stretches of DNA that form enhancer sequences. So if you come down here, here's those little guys. Your transcription factors called activators, and then on the other side, you have your DNA regulatory sequences called enhancers. And what these transcription factors will do, called activators, is they'll bend it over so that they can reach other transcription factors. So these are also transcription factors here. I'll circle them too. Okay, but they're not activators, but there are also other transcription factors here that help in the recruitment of RNA polymerase to locate and bind to the promoter. The abbreviation for polymerase is uppercase P O L. Okay, so that's the abbreviation for the term polymerase, POL. So what will happen is these other transcription factors will help recruit RNA polymerase, helping it locate a promoter. But these little guys here, called activators that are bound to the silencer, they will bend the DNA over with the help of other proteins so that they can reach over to RNA polymerase. And to keep it simple, they basically give RNA polymerase a kick in the butt, telling it, I need you to work faster or more often. Thus, we call these enhancers. These DNA regulatory sequences are enhancers because with transcription factors called activators, these DNA regulatory sequences play a role in enhancing gene expression. Okay, so again, these DNA regulatory sequences called enhancers play a role in enhancing gene expression. Okay, by being bound by transcription factors called activators that in turn will increase the rate of RNA polymerase activity, thus increasing the rate of gene expression and therefore increasing the amount of protein produced. Now, what's not shown on this figure is the other group, and I'm still looking around for a good figure. But if the transcription factors being shown up here, if they had been repressors, then instead of saying enhancer, they would have written down silencer. So for eukaryotic cells, we don't use eukaryotic DNA, we don't use the term operator. Okay. For eukaryotic cells, there are DNA regulatory sequences called silencers. And they are located also near the promoter, just like enhancers. Except for DNA regulatory sequences called silencers, the group of transcription factors that bind to those are called repressors. And then the repressors will do much the same thing as the activators. They'll bind to regions called silencers instead of enhancers. The repressors will bend the DNA over so they can reach RNA polymerase, except now, okay, they are either slowing down the rate of RNA polymerase activity or they're altogether preventing RNA polymerase from initiating and performing transcription. So we would get rid of this arrow in that case. Okay, so you would just swap this out for silencer, you would swap this out for repressor proteins, and then you would get rid of this arrow to indicate, or you would put a little decrease symbol to indicate RNA polymerase is being needed. Okay. So this is just another figure showing you a little bit more here, and I'll let you look at that one. This is from a non-major textbook if it helps, but they're meant to be the similar figures. Okay. So what I want to go over last is gene expression in different cells. As I said, when a sperm fertilizes an egg, we have a fertilized egg that transforms into a diploid zygote cell. That diploid zygote cell has 23 pairs of chromosomes in it containing genetic information from mom and dad. That one diploid zygote cell will divide into many cells that form an embryo. In other words, it'll develop into embryonic cells. All these embryonic cells inherit the same 23 pairs of chromosomes containing the same genetic information. But for reasons not understood, some of those cells, okay, will undergo cell differentiation by way of regulating or controlling which genes will be expressed. Okay? So therefore, let me put it this way. For someone who's type 1 diabetic, okay, they still refer to it as an autoimmune disorder. A person's own body is destroying the only cells in the body that can produce insulin. These cells are pancreatic cells. They are cells of the pancreas. Okay, they're called beta cells. So pancreatic beta cells are the only cells that have the ability to turn on gene expression of the insulin gene, therefore producing insulin. In a type 1 diabetic, that person's own immune cells are attacking and killing those pancreatic beta cells. The gene itself, the insulin gene itself, you can still find it in the rest of your somatic cells that form your body. But these other cells don't have the ability to activate gene expression of the insulin gene in them. and that's tied in partly to these transcription factors. So this is what I'm getting to. So the last of your notes, okay, has to do with gene expression, genes that are expressed by different cells. There's a group of genes that I don't know if they still use this term, but I do. There's a group of genes that are expressed by all somatic cells of your body. And they're expressed all the time for many of these. We call these housekeeping genes. Okay, sometimes they'll refer to housekeeping genes as genes that are constitutively expressed. That means they are expressed always. They are always on. The cell does not want to turn these off. We call these housekeeping genes. Examples of housekeeping genes would be any gene that codes for an enzyme involved in any one of the 3 stages of cellular respiration, such as glycolysis. It doesn't matter if it's a pancreatic cell, okay, a cell of the lens that's in the embryonic stage, or a nerve cell. So in other words, it doesn't matter if it's a developing embryo all the way up to an adult. It doesn't matter what type of cell it is. All cells need energy in the form of ATP, and they constantly need that energy. Okay. The protein enzymes responsible for catalyzing the chemical reactions of all 3 stages of cellular respiration, glycolysis, citric acid cycle, and oxidative phosphorylation. Those proteins always have to be present and working, but they're like automobiles. They can only work for so long, they have to be replaced. Therefore, the cell's always making these proteins to replace pre-existing ones that wear out. Therefore, any enzyme involved in catalyzing the first stage of cellular respiration, glycolysis, those genes that code for those enzymes, they are constitutively expressed. They are always expressed. Therefore, we call them housekeeping genes. And you can see here, down here, the rectangle represents the gene. Coloring it in green represents the gene as active or being expressed. Skip this one here. This one doesn't matter right now. What I'm going to go to next is this one. Some genes, for lack of a better term, I refer to them as cell-specific genes. Okay, and I already talked about it. Cells of the pancreas. 99% of the cells of the pancreas are exocrine cells. They're involved in producing and secreting digestive enzymes. Okay, roughly 1% of the cells of the pancreas cells are endocrine cells, called alpha and beta cells. Alpha cells produce a hormone called glucagon. And beta cells, as I said before, they produce a hormone called insulin. Beta cells are the only cells that are part of the pancreas that have the ability to activate this gene. But this is showing you, as I said before, every cell of your body, that's a somatic cell, definitely, okay, has the insulin gene in it. But only beta cells of the pancreas, only these specific cells have the ability to activate this gene, and part of it at least is tied into transcription factors. And again, they're still studying this. They're still trying to figure it out. Okay, they're showing you the hemoglobin gene here, but notice they don't list the red blood cell. Red blood cells are the only ones that can activate 2 genes, hemoglobin genes, one located on chromosome 16, one located on chromosome 11. Okay, the alpha and beta genes that code for the alpha and beta subunits. Okay, so they're showing you they're not expressed. Now, so you have some genes that are referred to as housekeeping genes. They are constitutively expressed. They are always expressed, and they're expressed by every cell of the body because they code for proteins that are always needed by the cell. There are other genes that are cell-specific genes. Only certain cells, such as beta cells of the pancreas, have the ability to activate certain genes, such as the insulin gene. So you find it in other cells of your body, but only those cells can turn it on. And then there are genes that are expressed only when needed, such as during cell division. Okay. This here, all of them in green, and notice these 2 are the same name. And this here. Okay. What I've tried to put a box around, okay, are 3 general types of proteins involved in promoting cell division, involving mitosis. Okay, we didn't go over this, but when I taught advanced cell and molecular biology, we did go over this. And if you pay attention to cancer drugs that come out, you might hear this once in a while. I keep forgetting the name of the drug. I have it somewhere. Okay, but roughly about 4 or 5 years ago, there was a cancer drug that came out, and they said this cancer drug is a cyclin CDK inhibitor. Well, this is what they're referring to. Notice Even though the first part of their names are different, the last part of their names are the same. Cyclin CDKs. Cyclin CDKs are responsible for promoting or causing cell division to happen, but they're only expressed when needed during cell division. So it's twofold. In other words, there's your third group of genes that are only expressed when needed, such as your genes that code for these cyclin CDK growth factors, proteins. Okay? But even when gene expression occurs when needed, that doesn't mean that all the genes involved in a certain cellular process, such as cell division, will be expressed at the same time. So when needed, a cell will express the genetic information that codes for G1 cyclin CDK. Then eventually, gene expression of this gene will be turned off when enough active protein has been produced to do its job. Then the protein will be cleared out, and another set of genes will be turned on that leads to the formation of a different active protein called S-phase cyclin CDK. S-phase cyclin CDK is the key protein in DNA replication during S-phase. But once DNA replication is completed, S-phase is completed, the genes that have been turned on that express and result in the production of this protein, well, gene expression of those genes is turned off and the existing protein is removed, while a third set of genes are being activated that code for mitotic cyclin CDKs. Mitotic cyclin CDKs help take the cell primarily through mitosis. But once that occurs, then gene expression of these genes is turned off, the protein is cleared out, one cell is divided into two. So that's the way I divide it up. Okay, so this third group of genes are genes that can be expressed by any cell of the body, but only when needed. So there are those genes which are turned on and then turned off. Then there are genes in certain types of cells that can never be turned on. Then there are genes that are cell-specific genes, such as beta cell, pancreatic beta cells, that will turn on and off the insulin gene, and they're the only ones that can do it. And then there are housekeeping genes. Every somatic cell of the body keeps these, or every cell of the body, let's just say every cell of the body, not only has not only turned on gene expression of these genes, but it stays on because the proteins they code for are always needed to do work that is always needed, such as the production of ATP. Okay. These last few figures are just review. Okay. So again, make sure that when you're going through chapter 17 that you understand from chapter 17 what you're going to need primarily, mostly, is an understanding of transcription, but you're also going to still need eventually an understanding of the process called translation. I'll stop the recording.